Welcome to this week's edition of Gene and Cell Therapy Updates, brought to you by Lucid Quest. Crystal Biotech has updated the label for Vyjuvek, a redosable topical COL7A1 gene therapy for dystrophic epidermolysis bullosa DEB. The new label expands its use to include DEB patients from birth and allows caregiver application with flexible dressing changes at home. This update, backed by real-world and open-label extension safety data, could improve access and adherence for DEB patients, enabling home-based therapy initiation and follow-up. In a Phase 1/2 trial, MYR-101, an intracranial gene therapy for Canavan disease showed early signs of success. In a cohort of 8 children, treatment with RAAV-OLIG001 ASPA demonstrated safety and functional improvements. After 12 months, cerebrospinal fluid N-acetylaspartate levels decreased significantly, myelination improved, and developmental milestones were met. These findings suggest target engagement and early functional benefits. Larger cohorts and longer-term data are needed to confirm results. Stanford University has introduced an AI assistant, CRISPR-GPT, designed to streamline CRISPR workflows. Built on a large language model, the tool designs and troubleshoots CRISPR experiments, predicts off-target effects, and adjusts for both beginners and experts. Early success stories highlight the tool's potential to speed up the design process and reduce errors, potentially accelerating gene therapy research. In a concerning development, Capsida Biotherapeutics has paused its Phase 1/2A trial of CAP002, a gene therapy for STXBP1 encephalopathy, following the death of the first dosed patient. The company has notified the FDA and initiated an investigation to determine the cause. The trial, running for about 2 months, is under review, with next steps to be determined after the investigation. Ocugen has entered into a licensing agreement with Kwangdong Pharmaceutical to commercialize OQ400, a modifier gene therapy for retinitis pigmentosa in South Korea. The deal includes up to $7.5 million in near-term payments, with additional milestones based on sales targets, as well as a 25% royalty on sales. The agreement leverages global data from Ocugen's ongoing trials, including a Phase 1/2 study showing improvements in low-light visual acuity. This partnership could expand OQ400's availability in the region and further support gene therapy development for eye diseases. The FDA has announced new restrictions on processing US patients' cells overseas. This regulatory shift may impact smaller cell and gene therapy companies that rely on international facilities for editing and reinfusion of patient cells. While the full scope has not been defined, the change is expected to disrupt global manufacturing chains, particularly for CAR-T therapies, and may lead to delays or increased costs for clinical trials using overseas processing. In Japan, the government has launched the Japan Expedited Approval Program to accelerate regenerative medicine and cell gene therapies. The program offers mentorship, market insights, and connections to local contract development and manufacturing organizations, contract research organizations, and pharmaceutical companies. This initiative aims to reduce drug approval timelines and modernize post-approval processes in Japan, increasing access to innovative therapies in the region. CHA Biotech, a South Korean company, aims to reduce the cost of cell and gene therapies by tenfold. By leveraging local supply chains and intellectual property strategies, CHA intends to drive down manufacturing costs, making these therapies more accessible to a wider population. These efforts could position South Korea as a major global player in the gene therapy market. Cook Myocyte, in collaboration with Veristat, has reached a Phase 3 milestone in its trial of iltemiocel, a cell therapy for chronic fecal incontinence in women with obstetric anal sphincter injury history. The two-stage, randomized, controlled study has enrolled over 100 participants across 26 active sites. With more than 95% adherence to the treatment protocol, the milestone marks significant progress in addressing this unmet need in women's health. Finally, researchers at the University of Zurich have demonstrated that stem cells derived from human-induced pluripotent stem cells can restore motor function after a stroke in mice. Transplanted neural stem cells survived, became neurons, and facilitated vessel growth and reduced inflammation. These results offer promise for future therapies aimed at repairing brain function post-stroke, though further work on delivery methods and safety switch mechanisms is needed. Stay ahead in gene and cell therapy research. Like, share, and subscribe for weekly updates. Visit lqventures.com or email us at info@lqventures.com for expert healthcare consulting. See you next time.